Take it away.
Hi, everyone. Good morning. Thanks for the conveners and thanks for being here. Today, I'm going to talk about mycorrhizal fungi and Earth system models. So, in the first place, why do climate scientists care about fungi at all? And luckily, we had Trevor and Ashley talking about the global carbon budget, so I don't have to spend too much time there. But pretty much, we want to know if the land surface will remain a carbon sink by the end of this century. And as you may know, most plants, terrestrial plants, associate with fungi, and they are responsible for a large chunk of the nutrient acquisition via these plants. So in this study that we published last year, we account for nitrogen and phosphorus limitation and the carbon cost of acquisition of those. And when we account for those two nutrients, we decrease global NPP in 50%. We ran E3SM, the Earth System model of the Department of Energy, with the FUN model on the fixation uptake of nutrients model, accounting for things like mineralization of nutrients and etc. And this result is in alignment with previous studies looking at CIMEP5 models that Will Witter published in the past. So how do we use resource optimization theory to calculate nutrient uptake in Earth system models? So we start from the principle that plants don't get nutrients for free. And so when we combine the FUN model with any land surface model, when we calculate net primary production before we allocate that carbon to growth, we will spend that carbon to acquire nutrients. And each row there has a different uptake pathway. And here I'm going to focus on the mycorrhizal fungi. And we represent in the FUN model two types of mycorrhizae, the arbuscular mycorrhizae and the ectomycorrhizal fungi. And just because these two are the most common mycorrhizal types, and they are substantially different in their physiology and the way they acquire nutrients. But pretty much the equation that governs that nutrient acquisition, or the cost of that nutrient acquisition, is the amount of soil nitrogen concentration in the soil and the root biomass. So the more nitrogen we have and the more roots we have, the cheaper it will be to acquire the same nitrogen. But we have a question about those K parameters in the cost functions. And so in the FUN model, the two types of mycorrhizae cost parameters were adjusted to mirror previous evidence of latitudinal shifts in the abundance of each mycorrhizal type and fertility gradients that we thought we were pretty sure about. And by doing so, we get that mycorrhizae is more competitive at low levels of soil nitrogen, which is in agreement with observations. However, we have a bunch of uncertainty when we do something like that. We have a lack of process understanding, so we don't know exactly what governs the nutrient acquisition. Is it biomass, really, root biomass, or is it root surface area, for example? We have a lack of metrics for model evaluation. As you may know, nutrient experiments are very limited. And we have also a lack of observational evidence about the mycorrhizal spatial distribution. So in this paper here that we published a couple of years ago, we analyzed 4 global maps of mycorrhizal distribution, and each map represents the fraction of ectomycorrhizae. The bottom figure shows the standard deviation between those 4 maps, and we can see that they disagree substantially. They disagree up to 50% over 40% of the land. So we're not sure where those mycorrhizae are. So what we did was we developed a global map of these cost parameters that I mentioned before for 2 types of mycorrhizae using empirical evidence of NPP allocated to mycorrhizae combined with MODIS data. So this paper here published by Hawkins et al. Shows that using in situ field and pot experiments showing the percentage of carbon allocation to each mycorrhizal type. And so we got those values and combined with MODIS NPP and a previous mycorrhizae map published by Nadia Sodilovskaya. And we combined that with soil grids, soil nitrogen content globally. That it's a product that was derived from machine learning using more than 200,000 observations, and maps of arbuscular and ectomycorrhizae fine root biomass that was also created using machine learning maps. So now, if you remember that equation, we would have 3 knowns and 2 unknowns. And what we did was to use minimization methods, downhill simplex, to find what was those cost parameters based on observations. We then compare those observation-based cost parameters, which is actually in the fun model in CLM. And when we compare the difference between these fixed cost parameters to mimic previous ideas of the cost of acquisition with the actual observation base, we see a large difference. So for the cost parameter associated to nitrogen, we overestimate, CLM overestimates it by 2 orders of magnitude. And when we look into the cost of acquisition related to carbon, we underestimate that cost for arbuscular mycorrhizae, and it's highly variable for ectomycorrhizae. So we can see that we usually underestimate those costs over arid regions in the world. We can also ask the question in the same way, but instead of looking at a map, we look at these plots versus plant functional types in CLM. So what we see is that for the arbuscular mycorrhizae carbon costs, KC is underestimated by CLM. And for the ECM, for the ectomycorrhizae, it's overestimated. So what we did now was to modify CLM5 to account for these observation-based cost parameters. And we perform a biogeochemical land model runs with CLM for 15 years using present-day climate and present CO2 levels. And we can see here the results of the amount of nitrogen acquired by these 2 mycorrhizal types with the new cost parameters. And we can see that now the model is predicting that ectomycorrhizae is getting much more nitrogen than before, but that the ectomycorrhizae is getting more nitrogen in the tropics, less nitrogen in the extratropics. So what we did then was the outcome of these new cost parameters was that it became overall cheaper to acquire nitrogen via mycorrhizae. And so what ended up doing is that CLM acquired twice more nitrogen as previous thought via the mycorrhizal uptake. When we did, so then is it true? Does it make sense at all? So we performed some comparisons with 3 variables. So we looked into modus albedo, FluxCom latent heat, and FluxCom GPP. And we see that the control run, so the previous thought cost parameters, behave worse than the observation-based cost parameters. So we increased the agreement with 3 variables when we account for the observation-based cost parameters. So we saw some improvements through the carbon, energy, and water cycles. And so finally, the real question is, when we see the improvement of this performance using these new parameters, it could be just a parametric coincidence, right? But I think like the final message is that what is CLM or other land models doing with the nitrogen, right? So when we look here at the control run, we see that the original model had a GPP of 121 petagrams of carbon per year. And when we now use the observed value, we get to the magical 123 FluxCom number for GPP. And so when we add this twice more nitrogen into the system, photosynthesis go up by roughly 2.2 petagrams of carbon. But in contrast, autotrophic respiration go up more. So it goes up 3.3 petagrams of carbon. So what we would think that getting more nitrogen would make the land more productive, in turn make the land a weaker sink. And that is because autotrophic respiration in CLM is highly dependent on the CN ratio. And so if the CN ratio goes up, autotrophic respiration goes up. I think I'll stop here, but there are lots of questions about how we allocate the nutrients in these models and how we can use new sources of information to learn more about the uncertainties that we have in the process understanding in these models, but also what we do with the carbon later on. How do we allocate that? Thank you very much.